As a result, a heterogeneous viral population containing viral variants able to withstand immune and drug pressure evolves to perpetuate the infection, but this adapted population is generally less virulent. Thus, generation of viral variants is key to HIV's success, though it comes at the price of reduced infectivity. Following penetration to infect target cells, further viral variants are generated by errors committed by the viral reverse transcriptase when it replicates the HIV genome. Precise replication of the viral genome requires that adenosine A, thymidine T, guanosine G, and cytidine C be incorporated into the growing DNA strand following the template genomic RNA. A pairing only with T and C pairing only with G, respectively.
Okay, I think we're ready to really test your knowledge of what we learned in this lesson. There are a lot of really awesome concepts that we've learned really in all the lessons up to this point, and we can start building an entire framework around how to understand particular traits. Let's use an example that combines the concepts of mutation, selection, and allelic frequency. The Ccr5 gene is located on human chromosome 3, and it produces a protein, the Ccr5 protein, that you can see here in green, and it resides on the surface of T cells. T cells are immune cells that fight infection. Now, although T cells do a pretty good job at fighting infection, in fact, they do an amazing job really, there's a virus out there that can really take them out. It's called HIV, or the human immunodeficiency virus. You might have heard of it before. The HIV virus binds really well to this protein called Ccr5. And when it does, the whole complex gets internalized. And this is how HIV gets inside T cells. It hijacks this protein and uses it to get inside the T cell, infect it, and then infect the entire immune system. So if you have a normally functioning Ccr5 allele, then you're at risk for HIV infection. However, there's an allele of Ccr5 that creates a very dysfunctional version of the protein, rendering it pretty much non-functional, and it can't even get to the cell surface of T cells. Without that Ccr5 protein, the HIV simply can't get in the T cells. The T cells are thus resistant to HIV infection. So here's your first question using this example. Is the mutation for the dysfunctional protein that makes the T cell HIV resistant more likely in the coding or non-coding DNA of the genome? I want you to check one of these boxes and then explain why you think that's the answer in this text box here. I want you to save your answer because guess what? You're going to need it in the next video.
You may wonder why we use the gene surgery to prevent HIV. Why not another lethal disease? Two reasons. First, safety. Second, real-world medical value. As a scientist and the father of 2 girls, safety is my #1 concern above all else. The gene we choose, Ccr5, is one of the best studied genes. In fact, about 100 million people naturally has genetic variations that disable Ccr5, protecting them against HIV. They were healthy. Our surgery caught exactly at the start of that natural variation. Decades of clinical trials has tested medicine to mimic that natural variation, including the first gene editing trial in the United States. Well, it proved safe. Also, protecting against HIV requires the simplest gene surgery, which is to remove just a few DNA letters. Correcting a broken gene, while helpful for more diseases like familial cancer or muscular dystrophy, it's more complex. Studies in the US hasn't shown this works well in embryos yet. The second reason we end up choosing HIV as our first target is the real-world medical value. HIV remains a deadly and devastating infection that needs medical advances. About 1 million people died of HIV last year. New infections remain 3 times higher than the UN AIDS target for 2020. No vaccine or cure exists. Taking pills daily from life carry healthy risks. In many countries, discrimination deepens the devastation of infection. Employer might fire people after discovering their HIV status. Doctor may refuse to treat. Ccr5 surgery, while not applicable to the general public, it may be valuable to help a few affected and very high-risk families protect their children from the same fate. This led to the National Academics in 2017 named vaccine-like disease prevention as an ethical application of gene editing.
Yeah, so I think there's a few issues, and one of them is obviously you need to be able to do this with a pretty high level of activity. So you need to identify a lot of cells, are able to effectively edit their genes in a substantial proportion, and then put them back in and hope that they ultimately take over. Even though we know that these people have not been blasted with chemotherapy to wipe out their own cell population. So I think some of the obstacles is creating a situation where this can be done effectively and efficiently so that you have a large proportion of cells that have been altered that can then be reinfused. Or if you can come up with a way to actually do it in the person, or to put them into stem cells that can be then transfused into the person and hopefully then continue to expand so that their own cells are increasingly missing the Ccr5. So I think that those are going to be the obstacles. There's been some clinical data with some of these strategies that have suggested little hints of promise, but nowhere close to a cure based on what's been tested so far. I think the other obstacle, in addition to creating a system that's extremely efficient and proving that it actually makes a difference, the other obstacle that everybody is struggling with a little bit is safety. You know, altering somebody's genes could potentially pose some risks, especially if there's some off-target alterations that occur and that you don't just hit the Ccr5 gene, but you perhaps hit other genes that might put people at risk for complications down the road. So that this is going to be looked at very carefully, will need to be regulated And studies will have to start small and get big if they demonstrate evidence of efficacy with long-term follow-up for safety. Not our traditional studies where you do it for a year and see if everything's okay, because the consequences of editing someone's genes may not become apparent for many years. Yeah, I would say that's exactly the case, is that this is very, very early in the pursuit of this type of treatment strategy in patients, with the goal mostly being trying to cure people or at least put them in remission for HIV so that they don't need to remain on antiretroviral therapy. And the bar is really high. You know, I think this is an extremely important avenue of research to try to cure people. But again, we have to balance whatever the risks and costs are against what are current strategies for managing HIV, and that's often one pill a day with no side effects and a chance for, you know, a long, healthy, normal life.